to another screencast. This is about fermentation or anaerobic respiration. And you want to describe or explain, first of all, the oxidizing agent of glycolysis. This will be helpful if you are able to identify that. Then definition of fermentation, the purpose of fermentation, the two types of fermentation, the reactants and the products of fermentation, and an example of an organism that would do each type of those subcategories of fermentation. So, title of the slide up here: fermentation produces organic molecules, including alcohol and lactic acid, recycles NAD+ to glycolysis, and occurs in the absence of oxygen. So in the absence of oxygen, this would make this process anaerobic. So this is sometimes referred to as anaerobic respiration. So anaerobic respiration. Notice that the fermentation definition up here does not include anything about making ATP, because fermentation itself does not make ATP. Fermentation, in the absence of oxygen or when oxygen levels are lower, will make organic molecules, will recycle NAD+. This is a key thing to look at right there. Recycles NAD+ to glycolysis and occurs in the absence of oxygen. So the image that we have here, we're going to start this fermentation pathway with the glucose coming into the cell. These several sets of arrows right here are supposed to represent glycolysis. And we generate then pyruvate. Now, in the cell, if oxygen is present, then that pyruvate will go down an aerobic pathway. So aerobic respiration, right? The pyruvate goes into the mitochondrion, right? Acetyl-CoA, Krebs cycle, electron transport chain, oxidative phosphorylation will happen. This screencast is going to be looking at when there is no O2, there's no oxygen present. So if there's no oxygen present, we can't go down this pathway over here. Or, in addition to, there's another way of thinking this would be that there's not enough O2, right? So in which case we are doing this aerobic respiration, but then we're going to add this. So when there's not enough, right, meaning that you're doing aerobic, right, and you are you're going to add the anaerobic pathway. So in fermentation, we're going to take a pyruvate, and it's going to be converted to either ethanol or lactate or lactic acid. So pyruvate to ethanol or lactic acid in the absence of oxygen or not enough O2. And this is all happening out here in the cytoplasm. So let's take a look at the specific details of this. There are two flavors, right? First, we'll deal with the lactic acid fermentation. So in lactic acid fermentation is going to reduce pyruvate to lactic acid. It's going to oxidize NADH to NAD+ and enables the ATP production via glycolysis. This is important, resulting in a rapid production of ATP, but in a small quantity. So again, here's glucose. These arrows right here represent glycolysis. We're not talking about that process. We're talking about taking this pyruvate and converting it into lactic acid. So if you look at this diagram, or this part of the diagram over here in the cytoplasm, this is what's happening. Glucose, glycolysis, generating pyruvate. The bottom of this diagram, this is the fermentation pathway right down here. This upper part here is glycolysis, not fermentation. The fermentation pathway is down here. This pyruvate molecule, 3 carbons, we are going to come up here, we're going to reduce it. When you reduce pyruvate, you end up getting lactate or lactic acid. This is the byproduct. This is not why you're doing this fermentation pathway, right? This is the byproduct. So if I'm going to reduce pyruvate, something else has to be oxidized. Well, looking at this, what's going to be oxidized? The answer is NADH. There's NADHs right here that are going to be oxidized. So this is an oxidation. This is the reduction. And when I oxidize NADH, I make NAD+. This is important. This is the important part of the fermentation pathway right here, is that the NADH is being oxidized to regenerate this NAD+, because with NAD+, I can now run glycolysis. Right? I can keep glycolysis running. Now, in an aerobic situation, this pyruvate is moving on, right? And the oxidative phosphorylation, right? Electron transport oxidative phosphorylation is doing this, is going to take NADH and oxidize it, and you're going to get NAD+. This NAD+ could then come back and be used in glycolysis, right? So aerobically, in an aerobic situation, this oxidation is regenerating NAD+ to be used in glycolysis. But anaerobically, right, this is not happening over here, right, or is not happening fast enough. You're doing this the maximum as you can. It's not happening fast enough, so then you add this situation over here. You're going to then reduce pyruvate to lactic acid and then oxidize this NADH, regenerate NAD+. So this, now going back up and looking at the title here, makes maybe a little bit more sense. Lactic acid fermentation reduces pyruvate to lactic acid, oxidizes NADH to NAD+, enables ATP production via glycolysis. Here's glycolysis, ATP production, resulting in a rapid production of ATP, but in a small quantity. So you get this, but then the price you pay is that the lactic acid concentrations begin to build up, right? The cell is not, quote unquote, trying to make lactic acid, right? It's trying to, quote unquote, make this NAD+ here to keep glycolysis running. So organisms that do this, lactic acid fermentation, would be things like animals, right? Animal cells, right? For example, your muscle cells. So muscle cells that are contracting using a lot of ATP, right, will, in times that you're not getting, this pathway is not running enough, right? It's running as fast as it can, but the demand for ATP is going way up. You're exercising your muscles, right? So then your cells then add, right, this lactic acid fermentation part here to make even more ATP to keep those muscles contracting. But the problem is that you're building up lactic acid there, and the muscles don't like that, and that becomes painful, and muscles eventually shut down. The other pathway, the other fermentation pathway here is ethanol or alcoholic fermentation. Notice that this is very similar to the other one, right? So let me just flip back to the other, right? There's lactic acid, there's ethanol, right? or alcoholic fermentation. There's an extra step right here in this, right, where the pyruvate is first going to be decarboxylated. This is a deeper understanding right here. If you can understand that if I decarboxylate, right, that is, I'm removing a carboxyl group, and here's the carboxyl group, an old friend that you forgot, right? And if I decarboxylate and take this hydrogen off, if I take that hydrogen off, that is oxidizing. So if I oxidize this and decarboxylate, oxidize and decarboxylate, oxidize would be take the hydrogen off, that goes away, and what am I left with? Oh, I'm left with CO2 right there. So I oxidize and decarboxylate in that step, and then I'm going to reduce acetaldehyde to ethanol. This is a reduction. Well, if there's a reduction happening, there's got to be an oxidation happening somewhere. Here's the oxidation. We're going to oxidize NADH to NAD+ right here. And so this fermentation pathway is similar to, but different than, the lactic acid fermentation pathway. In this step, in alcoholic fermentation, we have this extra step right here, this oxidative decarboxylation, to get this to this acetaldehyde thing, which is then going to be reduced to ethanol, NADH, oxidized NAD+ generated. And again, this is the key point right here, is that this fermentation pathway is providing NAD+ so that glycolysis can keep running. If I can keep running glycolysis, I can keep making ATP cheaply. Right? This happens when there's no oxygen present. So this whole side of the diagram is not happening over here. Right? So organisms that are able to do this are any sort of anaerobic bacteria, and they're making enough ATP to keep their cells running. Right? Another classic example would be yeast. Right? Yeast is a unicellular fungus, and it is a eukaryote. Right? So you have prokaryotes and you have eukaryotes, eukaryotes that are able to do this alcoholic fermentation. Right? Yeast, incidentally, are able also to do aerobic respiration. So they're pretty cool. They can actually survive in anaerobic situations, right? Which is the whole process of fermenting grapes or fermenting barley, fermenting rye, and you end up getting then ethanol. Again, that's the byproduct right here. This is not the point. Right? For the cells are trying to do this, regenerate this, but we as humans have been able to say, oh, okay, look, they can also make this ethanol here, and we're going to utilize that and make things like beer and wine and what have you from that. So kind of interesting little connection there, but again, going back to the cellular energetics part here, fermentation pathways enable glycolysis to keep going. So go up here to this title, alcoholic fermentation decarboxylates, right here, and reduces, right here, pyruvate to ethanol, oxidizes NADH to NAD+, which enables glycolysis to keep running, resulting in rapid ATP production, but in a small quantity. Okay. Graphically organizing all of that, skipping to the end here. Here we go. Anaerobic respiration, there are two types. And again, you're focusing probably on the bottom half of this diagram. The top half here will give you a little tip here. So this is going to be glycolysis up here. So then what do you get? You have here, and then there's something happening on the left side or something happening on the right side, right? So what are the two types? What are the reactants? What are the products? Where is this happening? And what is the purpose of this overall summary there? Okay. Hope that's helpful. Bring your questions to class.